All right, you're welcome to my class. This is PSB 203, Introductory Plant Biotechnology. So, I'm going to talk about the historical development of plant biotechnology. Definition of plant biotechnology. Now, what are plants? Plants are basically multicellular organisms. They have chlorophyll. They can reproduce either through spores or seeds. They are the primary producers in the food chain. They are living, of course, and they are found in the kingdom Plantae. So that is for plants. So biotechnology is also known as plant genetic engineering. Please take note of this important definition or fact. They are also called it's also called plant genetic modification. So when I say plant biotechnology, plant genetic engineering, plant genetic modification, I'm saying the same thing. It's a branch of biotechnology that involves the manipulation of plants genetics to create desirable traits or improve the plant's characteristics for various purposes. I'm sure you've heard about DNA during paternity tests. So if plants are living organisms too, therefore means that they have DNA. And so when we say plant engineering, we are just manipulating the contents in the DNA of the plants to produce what we want. For example, if I want a plant to be very, very big, fruit wise, like I want the fruits from the plant to be very, very big and sweet, I will manipulate the DNA to produce that kind of result for me. If I want my plant or my crop to be resistant to diseases or to environmental stress, I will manipulate the DNA to give me that result. So when scientists manipulate plants to produce desirable traits or results, it's called plant biotechnology. And so it could be traditional, which means those days people used to manipulate plants' DNA, maybe by crossing different varieties of plants and all of that. So we call it traditional plant biotechnology. Or it could be modern, going to the lab, extracting the DNA, you know, recombining them with another form of DNA, transferring them back again to the plant. All of this could be termed modern plant biotechnology. So, plant biotechnology could be traditional or modern. Okay. It allows scientists to introduce specific genes into plants, modifying existing genes or removing undesirable traits. Apart from bringing in new traits, you can also remove undesirable traits. For example, food getting spoiled within a short time, let's say the tomato plant, for example, is an undesirable trait. And so you say, I don't want my tomato to get spoiled, so I'm going to remove that gene responsible for it, or something like that. And it becomes biotechnology. So, some aspects of plant biotechnology we're going to be looking at is the genetic modification, tissue culture and cloning, genome editing, marker assisted selection, molecular farming, plant disease diagnostics, conservation of plant genetic resources, and biofortification. It's very important for us to know all these facts. I always tell my students when I'm teaching them history, you don't need to memorize everything, but just know draw out the important facts. For example, know what happened in 1970, know what happened in this particular year. Even if it's just one fact you want to take from that paragraph, it's important. So, the origin of plant biotechnology can be traced back to the early 20th century. Researchers started researching in that area in the early 20th century. So, if I were you, just jot down in your notes, it started in the 20th century. Full stop. You don't need to write heaven and earth. Then you go to the next line of your notes. It was in the 1970s that the foundation of modern biotechnology was laid. One of the significant breakthrough was the development of, so you just write 1970, development of recombinant DNA technology. That's all you pick from that paragraph. Okay, please go with me. So in 1983, what happened? A bacterial gene was introduced to tobacco plants. You just write in the next paragraph, 1983, bacterial gene introduced to tobacco plants. So there are many stories again. This breakthrough paved the way for further advancement. Plant biotechnology, just you continue. Plant biotechnology contributes to development of. So that's another important fact. The fourth and the last point on this page. Plant biotechnology contributed to the development of genetically modified crops. Now you're having GM crops. So what is the meaning of GM? Genetically modified. So if I say a GM rice is different from the normal rice you are eating, I'm telling you that that rice has been genetically modified. That means the genes, the DNA of that rice has been manipulated to produce a desired result. And that is plant biotechnology for you. Okay? So let's pick what happened in 1983. So just pick all these important facts. And it's still about history of plant biotechnology. I that the first one was the origin, but now I'm delving into going deeper into the history. We've said some of these things before, but I just want you to take note of them. For example, in 1990s, plant biotechnology became more pronounced in the science world. So you can say in the 1990s, more work was done and all of that. In the 1970s, recombinant. Okay, I think we said that before. And then you come to the 2000s. In 1994, I think we've said something about the GM plan, but listen to this one. It's very important. In 1994, the Flavor I think FLAVRSAVR tomato was produced. This tomato can last very long. You don't need to put it in the fridge. There's a gene that is responsible for breaking down the cell wall of the tomato plant or tomato fruit that will cause it to go bad or be spoiled. So, if that gene is silenced, that means the gene is maxed, it's prevented from being expressed. You are going to learn all these things later, gene expression, all of that. But just imagine, for example, we know that time for if someone can delay time, you know that time is like there's 60 seconds make 1 minute and all of that. And so somehow you create a time machine and then you have the power to delay time. You can then do a lot of things within 5 seconds. Just imagine something like that. So kind of there's a gene responsible for causing the tomato plant to be bad, and now you are silencing that gene. It's not as if it's not going to go bad, it's going to go back eventually, but it's going to take long, or it's going to take longer for it to go bad. And so you have succeeded in creating a new GM tomato. They call it the FLAVRSAVR tomato, and it was created in when 1994. Please take note of that. And so you need to know what happened in the year 2000s, marker assisted selection, and all of that. Marker assisted selection, um, imagine people planting palm trees, and there are different varieties of palm trees, but you will not know which is which until you allow them to grow and start producing fruits. But because of this marker assisted selection, you can know the variety of these different seeds of palm trees, even from the seed level. You don't have to plant it and wait for 15 years. You now say, this one is the one that is sweet, this one is the one that is big. No. Using marker assisted selection, you'll be able to differentiate from the seed level. And that technology started in the year 2000s. Okay. RNA interference technology is harnessed to silence specific genes. So it was RNA interference technology that was used to silence, I think, specific genes. And so pick out all the important facts from this page. You can screenshot it, you can jot that information in your notes, okay? So what happened in the 2010s? Molecular farming gains attention as plants are engineered. Okay, you must have heard something about the insulin corn. There's a maize that has insulin gene implanted into it. So instead of going to the hospital for diabetic patients to have insulin injection, now they have succeeded in incorporating the insulin gene into a corn, a maize. So diabetic patients can start eating insulin corn, for example. That is molecular farming. You are using plants to produce important pharmaceutical proteins and vaccines. Imagine the coronavirus vaccine, something like that, being incorporated into plants. And in the year 2000s, plant biotechnology continued to evolve around the continents, around basically everywhere, and new varieties of crops and all of that. So that's the end of my video. I hope it's not that long. It's 11 minutes, I think. Please stay tuned to the next for the next video. And if you have any questions, I'll be very, very happy to respond to them via the comment section. Please do subscribe to my channel and God bless you. Thank you.